In the latest issue of Trends in Molecular Medicine, we present an article, Beta Cell Fragility as a Common Underlying Risk Factor in Type 1 and Type 2 Diabetes. This is Adrian Liston on behalf of the authors. Glucose needs to be maintained in the bloodstream at a very steady level. However, glucose does not enter the bloodstream at a very steady state. Instead, we have a large amount of glucose that enters our bloodstream after a meal. In order to compensate for this, the beta cells in pancreatic islets are able to sense the high amount of blood glucose and release insulin. This insulin then circulates to different organs, most importantly the liver and skeletal muscle, and it signals these organs to scavenge excessive blood glucose out of circulation and store it for a later time point. This allows the blood glucose levels to rapidly return to normal. Defects in blood glucose control can occur in multiple different ways. The two most important are autoimmunity and insulin resistance. In the case of autoimmunity, the initiation of an autoimmune reaction against insulin and other antigens that are expressed by beta cells in the pancreatic islets causes a destruction of these cells. This means that after the entry of high amounts of blood glucose, the beta cells are not present in order to release insulin, which means organs such as the liver do not respond to the high levels of glucose, and a high level of circulating blood glucose is maintained for a pathogenic period of time. This can result in type 1 diabetes. In the second pathway, organs such as the liver and skeletal muscle can become resistant to insulin signaling. This means that even following the release of insulin from beta cells in the islets, that insulin is not promoting a scavenging of the blood glucose to a good enough extent. This means that blood glucose levels can stay relatively high for a prolonged period of time, causing pathogenic outcomes. This process can evolve into type 2 diabetes. In the current paper, we present a model for beta cell fragility underlying both type 1 and type 2 diabetes. It is known that multiple different inputs can cause a beta cell to fail. These inputs can be autoimmune, inflammatory, glucotoxicity, oxidative stress, and unfolded protein stress. While each of these processes have very different origins, in each case, the beta cell under this stress will start to fail. It can fail by undergoing apoptosis, having functional impairment, undergo senescence, or differentiate into a state where it is no longer able to produce insulin. In the beta cell fragility model, we propose that there is variation from individual to individual in how fragile the beta cells are at integrating these negative signals into negative outputs. For example, beta cells that are considered to be fragile would be able to undergo apoptosis at a very high rate, even with a very low amount of input from autoimmunity, inflammation, and the other negative inputs. This makes them highly sensitive and highly likely to undergo apoptosis and other negative functional changes. At the opposite end of the spectrum, we would have robust beta cells. These are beta cells that are quite resistant to any induction of apoptosis and other negative processes, which means they need to have a lot of negative input, such as inflammation or oxidative stress, before they would undergo even a little bit of apoptosis or dedifferentiation. This means that these beta cells are able to function long-term even with a moderate amount of negative input into the system. In the beta cell fragility model, this places fragile beta cells as being the commonality between both type 1 and type 2 diabetes. We can consider that the combination of fragile beta cells with autoimmune prone T cells would result in type 1 diabetes, as even a small amount of autoimmune pressure will be enough to kill these fragile beta cells. On the other hand, a combination of fragile beta cells with susceptibility to insulin resistance would be able to drive type 2 diabetes, because the insulin resistance causes an upregulation of insulin production in the beta cells, therefore driving cellular stress and death in the fragile beta cells. Now, the evidence for this model comes from a variety of different sources. In terms of human genetics, the association of GLIS3 with both type 1 and type 2 diabetes indicates a shared mechanism. As GLIS3 controls the susceptibility of islets to apoptosis induction, This indicates that apoptosis induction or beta cell fragility is indeed a common mechanism between type 1 and type 2 diabetes. Secondly, if we look at diabetes epidemiology, there are characteristics that are shared between type 1 and type 2 diabetes, which again supports a common mechanism underlying both diseases. Thirdly, animal models for diabetes indicate that beta cell fragility can be a factor. In particular, recent data from the NOD mouse model, the gold standard model for type 1 diabetes, indicates that variation in beta cell fragility or robustness drives susceptibility or resistance to diabetes. And finally, in vitro islet models can be used to mimic the genetic variants that are associated with type 2 and type 1 diabetes in humans, and these variants have been shown to drive apoptosis susceptibility or resistance in beta cells in vitro. As well as proposing the beta cell fragility model and outlining the evidence for and against this model, we pose several outstanding questions, which will be quite important for further development of this field over the next 5 to 10 years. First, we need to understand how much genetic overlap is there between type 1 and type 2 patient subpopulations that may allow us to identify particular sets of genes that are more likely to overlap between these diseases and possibly be driving the beta cell susceptibility phenotype that is associated with type 1 and type 2 diabetes. In addition, this model, if it can be validated, would have quite important therapeutic consequences. For example, dietary modification is currently used in type 2 diabetes, and yet this model would suggest that it may be useful to prevent at-risk children from developing type 1 diabetes. And of course, if we identify a new pathway to diabetes development, this constitutes a new target for therapeutic intervention. It is possible that the next class of anti-diabetogenic drugs will be those drugs that enhance the robustness of beta cells, therefore preventing beta cell apoptosis and diabetes development.